Bismillahirrahmanirrahim. Welcome to today's lesson on dietary lipid metabolism. Lipids are a heterogeneous group of water-insoluble hydrophobic organic molecules such as fatty acids, triacyglycerols, glycerophospholipids, steroids, and sphingoglycolipids. Because of their insolubility in aqueous solutions, body lipids are generally found compartmentalized, as in case of membrane-associated lipids or droplets of triacylglycerols and adipocytes, or transported in the blood in association with proteins, as in lipoprotein particles or on albumin. Lipids are a major source of energy for the body, and they also provide the hydrophobic barrier that permits partitioning of aqueous contents of cells and subcellular structures. Lipids serve additional functions in the body. For example, some fat-soluble vitamins have regulatory or coenzyme functions, and the prostaglandins and steroid hormones play major roles in the control of the body's homeostasis. Deficiencies or imbalances of lipid metabolism can lead to some of the major clinical problems encountered by physicians, such as atherosclerosis, diabetes, and obesity. Digestion. The average daily intake of lipids by U.S. adults is 78 grams, of which more than 90% is triacylglycerol tag, formerly called triglyceride, that consists of 3 fatty acids esterified to a glycerol backbone. The remainder of the dietary lipids consists primarily of cholesterol, cholesterol esters, phospholipids, and non-esterified free fatty acids. The digestion of dietary lipids begins in the stomach and is completed in the small intestine. Digestion in the stomach. Lipid digestion in the stomach is limited. It is catalyzed by lingual lipase that originates from glands at the back of the tongue and gastric lipase that is secreted by the gastric mucosa. Both enzymes are relatively acid stable with optimal pH values of 4 to 6. These acid lipases hydrolyze fatty acids from tag molecules, particularly those containing short or medium chain lengths, more than 12 or equal to 12 carbons. Fatty acids of short or medium chain length, such as are found in milk fat. Consequently, these lipases play a particularly important role in the lipid digestion in infants for whom milk fat is the primary source of calories. They also become important digestive enzymes in individuals with pancreatic insufficiency, such as those with cystic fibrosis. Lingual and gastric lipases aid these patients in degrading tag molecules, especially those with short to medium chain fatty acids, despite a near or complete absence of pancreatic lipase. Let's talk about cystic fibrosis. Cystic fibrosis is the most common lethal genetic disease in Caucasians of Northern European ancestry in the United States. Cystic fibrosis is an autosomal recessive disorder caused by mutations to the gene for the CF transmembrane conductance regulator protein, also known as CFTR protein, that functions as a chloride channel on the epithelium of the pancreas, lungs, testes, and sweat glands. Defective CFTR results in decreased secretion of chloride and increased uptake of sodium and water. In the pancreas, the depletion of water on the cell surface results in a thickened mucus that clogs the pancreatic ducts, preventing the pancreatic enzymes from reaching the intestine, thereby leading to pancreatic insufficiency. Treatment includes replacement of these enzymes and supplementation with fat-soluble vitamins. Cystic fibrosis also causes chronic lung infections with progressive pulmonary disease and male infertility. Next, we will discuss emulsification in the small intestine. The critical process of dietary lipid emulsification occurs in the duodenum. Emulsification increases the surface area for the hydrophobic lipid droplet so that the digestive enzymes which work at the interface of the droplet and the surrounding aqueous solution can act effectively. Emulsification is accomplished by 2 complementary mechanisms, namely the use of detergent properties of the conjugated bile salts and mechanical mixing due to peristalsis. Bile salts made in the liver and stored in the gallbladder are amphipathic derivatives of cholesterol. Conjugated bile salts consist of a hydroxylated sterol ring structure with a side chain to which a molecule of glycine or taurine is covalently attached by an amide linkage. These emulsifying agents interact with the dietary lipid droplets and the aqueous duodenal contents, thereby stabilizing the droplets as they become smaller from peristalsis and prevent them from coalescing. Degradation by the pancreatic enzymes. The dietary triacylglycerols, cholesterol esters, and phospholipids are enzymatically degraded in the small intestine by pancreatic enzymes whose secretion is hormonally controlled. So first we will discuss the triacylglycerol degradation. The tag molecules are too large to be taken up efficiently by the enterocytes of the intestinal villi. Therefore, they're hydrolyzed by an esterase called pancreatic lipase, which preferentially removes fatty acids at carbons 1 and 3. The primary products of hydrolysis are thus a mixture of 2-monoacylglycerol and 3 fatty acids. The pancreatic lipase is found in high concentrations in pancreatic secretions, and it is highly efficient catalytically, thus ensuring that only severe pancreatic deficiency, such as that seen in cystic fibrosis, results in significant malabsorption of fat. A second protein called the colipase is also secreted by the pancreas. It binds the lipase at a ratio of 1:1 and anchors it at the lipid-aqueous interface. Colipase restores activity to lipase in the presence of inhibitory substances like bile salts that bind the micelles. The colipase is secreted as the zymogen, the procolipase, which is activated in the intestine by trypsin. Orlistat is an anti-obesity drug which inhibits gastric and pancreatic lipases and thereby decreasing fat absorption and resulting in weight loss. Cholesterol ester degradation. Most dietary cholesterol is present in the free form, with 10 to 15% in the esterified form. Cholesterol esters are hydrolyzed by pancreatic cholesterol ester hydrolase, which is also known as cholesterol esterase, and that produces cholesterol plus a free fatty acid. Activity of this enzyme is greatly increased in the presence of bile salts. Next is phospholipid degradation. Phospholipid degradation involves phospholipase A2. Phospholipase enzyme needs trypsin for activation and bile salts for optimum activity. What happens in phospholipid degradation is that phospholipid is acted upon by a phospholipase A2 enzyme, which converts it to lysophospholipid. Then lysophospholipase enzyme acts on lysophospholipid and converts it to glyceryl phosphoride base, which can be excreted in the feces or may be absorbed and reused.